Hi, I'm Julie Berame, and to my co-researchers, Aris Lapada, Prosel Miguel, Liz Noguera, and Siba Alam. Today, I'm presenting the findings of our study entitled Micronucleus Evaluation in Exfoliated Human Buccal Epithelium Cells among AWIS workers in Payatas, Manila, Philippines. Mainly, the reason why we focus to conduct our study among AWIS recyclers because there are limited studies conducted in the Philippines on the harmful effects of AWIS materials to human health. The improper recycling of electronic waste by informal recyclers often leads to contamination of the environment and most importantly among humans' health. With this, we aim to assess risk of informal e-waste recyclers due to their direct exposure to e-waste toxicity and by examining the presence of micronuclei in buccal epithelium cells as indicator for health risk. Payatas has been a disposal site for the city's waste electrical and electronic equipment for over 2 decades. The management of e-waste becomes a major challenge due to the steady increased volumes of local e-waste in the recent past. The generation of tremendous amount of bulky e-waste containing variety of hazardous substances is currently a major social problem and threat to the environment and to human health. The increase of e-waste is unsustainable disposal and becomes major concern for environmentalists which cause several human body illnesses. So e-waste contains organic and inorganic compounds along with heavy metals and trace elements, and these pollutants can have adverse and negative effects on human health. In this study, we use descriptive statistics and regression for the data analysis. So as you seen in table, t-test and chi-square test were used to describe and associate two groups of samples. As seen in tables, there are differences in length of exposure, occupation, smoking, and alcohol drinking were highly significant. Those living and working in e-waste recycling sites had greater evidence of DNA damage than those living in controlled area. Three variables were highly significant, indicating that the two groups of exposed and unexposed participants differed in terms of their health conditions based on the findings of the study. As seen in Table 2, the difference between the control and exposed group was significant in terms of increase in the number of micronuclei. Same also in Table 3. Results showed that e-waste workers had increased micronuclei compared to the control group. So similar trends were also found in the linear regression analysis drawn from participants' length of e-waste exposure, indicating that as the length of e-waste exposed increased, the number of micronuclei found in the participants' buccal epithelium cells increased as well. So in conclusion, longer exposure to e-waste materials may induce genotoxic damage in human cells, which is a serious concern leading to adverse effects to human health. Thank you very much for your listening.